Today's talk is going to be by Zhavoronkov, the CEO and founder of Insilico Medicine, a biotechnology company pioneering a next generation AI and robotics platform for drug discovery. And he's the founder of Deep Longevity, and also he's a fellow at the Buck Institute. So I'll hand it over to Alex to continue the talk.
Thank you for this introduction, and I would like to thank Oxford Society for Aging and Longevity for this generous introduction. I am presenting from Hong Kong right now, so it's 2 a.m. and it's part of my life's mission to kind of spread the word about longevity biotechnology and educate. So we'll try to be as helpful as possible. For those of you who have not gotten a link to the conference and are logging in and using a code, congratulations, you're smarter than average already, because I know that the link has not been circulated, but the ID was circulated. And congrats on joining. I'm going to tell you a story today about dual purpose longevity therapeutics. Drugs that target aging and disease at the same time. I'll introduce Insilico Medicine a little bit and tell you about a few resources that are available for you to get deeper into the field, into longevity. And I wish I had this kind of lecture when I just started this journey about 20 years ago. Well, 19 and a half years ago. So here is the, and Salmana, maybe it's a good idea to also admit people from time to time because I see that they're popping up.
Yeah, no worries, I'll do it.
All right, so I don't need to worry about that. Here is a slide which pretty much tells you the story of Insilico. So we're a truly global company. Our mission is to extend healthy, productive longevity. Our main value is to ensure that the drugs reach the patients. So our main stakeholder is the patient. We are committed to relentless innovation, so we never stop developing new algorithms. And we're committed to transparency and integrity. So we try to publish most of our research openly, and we usually publish 1 to 2 research papers a month. We are a platform and a pipeline company. So platform meaning that we also provide AI software to the entire market. And I'm going to talk a little bit about that and show you a couple demos. Some of those platforms are so simple that high school students can use it, and they do. And we are also using this AI platform to discover our own therapeutics. And we have a pipeline of 31 therapeutic programs, 29 fully owned, and some are partnered. Many of those therapeutics have dual purpose, especially our first 2 lead programs. So a little bit about, so I have to make those disclaimers. So no forward-looking statements, not investment advice. And also, I must say that I am very, very committed to longevity research. So if you like to see my personal pledge, very committed to longevity research, and pretty much everything I earn, I put back into longevity biotechnology, mostly investing in companies and supporting nonprofit initiatives. Also, if you want to learn about longevity medicine from medical professionals, I highly recommend going to longevity.degree. It's a volunteer effort by physicians and scientists where you can get free courses. There are currently 3 courses out there, Longevity Medicine 101 and 201, where you can actually earn CME credits, continuing medical education credits, if you're a physician in the US. And you can get the basics of aging research and longevity medicine in the context of treating patients. So, by the way, not treating patients, but learning how to do clinical trials and work with patients who have questions, because nowadays patients are very informed. And if you are planning to pivot your career into medicine, that's a very good place to start. Also, another resource I want to tell you about, because again, the way we learn is in our hands, right? So now a lot of tools are available for free. One really cool lecture that I find to be especially useful when learning about longevity biotechnology is by Boston Consulting Group, Michael Ringel, the managing partner, managing director and senior partner at BCG, responsible for healthcare practice. He's been in the industry for 25 years, with BCG with 25 years. And he definitely knows a thing or two about strategy in pharma. And in this lecture, you can again find it easily on YouTube. He presents the emerging area of longevity biotechnology, does the landscaping of the different companies, different approaches, different business models. And if you're formulating your career right now going into biotech, it's a very good place to start because the time is now. Why do we go after aging in pharma? One main reason is because if we completely eliminate one individual disease, for example, cancer, it provides very marginal gain, marginal dividend in terms of life expectancy gain at the population scale. So, for example, there were many simulations, this is just one of them, in Aging Research Reviews 2006, showing that if you completely eliminate cancer, it gives you about 2.3 years at birth in terms of life expectancy, 1.2 years at age 65. So that's not exactly super significant because we've seen that COVID reduced life expectancy in the US much more dramatically than 1.2 years. So basically, no COVID equals defeat of cancer, and because something else is going to kill you. So it's very, very important to focus at the core fundamental aspects of aging to be able to reduce the probability of many of those diseases happening prematurely. So the idea is to die young, as old as possible, as young as possible, as far as possible into the future, and prevent some of those diseases. Most of the diseases are age-related, so it's a very good strategy to go after aging and disease at the same time. So in order to answer this question, what is the most credible and sustainable way to research and commercialize longevity? We established a conference. So I co-founded this conference originally in Switzerland, in Basel. That's where Roche and Novartis are headquartered. So it's the city of pharma, really the capital of pharmaceutical industry. And then it moved to the University of Copenhagen, and we're organizing it for the 10th year in a row. So next year it's going to be our 10th anniversary. This year we had more than 400 people on site, several thousand dialed in online, hybrid format. So I highly encourage you to go on this website and also look at the lectures, as we also publish the YouTube lectures for free, and it's a 5-day event. Pretty much every big name globally is presenting at this conference, including the many startups. And how can AI help solve aging? So, I started my career in computer science and then switched into biotechnology, focusing on AI. And I realized around 2013, 2014, that new approaches, next generation AI, deep generative reinforcement learning, allows you to go at after pretty much every area of biomedical sciences with a pharmaceutical focus. So you can utilize deep learning and deep generative reinforcement learning for biological age prediction and monitoring, for generation of synthetic data, using age as a generation condition. We can use it for mental health, depression, motivation, behavioral modification. So we just recently published several papers on psychology of aging. I'm not going to touch on that. And a company called Deep Longevity is focusing on that technology. So applying deep learning for biological age prediction and mental health and behavioral modification, predominantly using deep neural networks to predict your chronological age in a healthy state and correlate that with the different mental states and psychiatric diseases. And Insilico focuses on many other things. So of course, it also uses age prediction for target identification, for biomarker development, for protein design, for molecular screening, molecular generation, uses deep generative reinforcement learning for precision medicine, expansion of the data sets from public sources. By the way, at Insilico, I must make a disclaimer, we don't use proprietary patient data. We always use data from the public repositories because there is over 1.5 to $2 trillion worth of government funding spent on over the past 35 years that we can use to train your AI. And again, it can help you go end to end in many areas of pharmaceutical sciences. So in 2014, the new technology called generative adversarial networks emerged, published originally by Ian Goodfellow at that time at the University of Montreal, working with Yoshua Bengio. Then he moved to Google, then Apple. Now he's at DeepMind. So he came up with this concept of generative AI, so AI imagination. And in 2017, those networks already got pretty good at generation of fake faces, so deepfakes. And in 2017, this paper was published by Karas showing that those people were generated by AI, but they do not exist. What is great about generative approaches is that they retain many features that are extremely biologically relevant. So you can see that these people actually do have age and sex in there, and many other features that you can derive from those faces, even though they don't belong to specific individuals. And again, the most important biological feature, age, is retained, and you can use predictive AI, so deep neural networks trained to predict age. You can actually use them to verify that age is present in those generated data sets. And around 2014, 2015, and 2016, I published a paper showing that you can use deep neural networks to predict chronological age in a healthy state. And we can use it, we can call it biological age if it has biological relevance. So pretty much any data that changes in time, like transcriptomics, proteomics, methylation, imaging, your facial features change in time with aging, EEG, ECG, activity, so all kinds of wearables can be used to predict your chronological age. You can use blood tests, and again, even psychological survey data to predict age using deep neural networks. As those deep neural networks get trained, they learn to generalize and learn the various features that are important for age prediction. And if you are utilizing several types of biological data, these networks learn biology, learn basic longitudinal changes that transpire in time. Very often those changes are so minute that you won't be able to recognize them as a human. So you need to have superhuman AI to be able to make those accurate guesses for age prediction. And then once those networks are trained, you can derive pathways, so signaling pathways, identify targets, create causal graphs. And as I mentioned, you can also use them to generate synthetic biological data. And since 2016, we published a number of aging clocks, predictors of age, using multiple data types. So just as a human would, you can predict the person's age using imaging data, so photographs, right? Behavioral aging, so the way the person behaves. So think of AI as another human. So if the human can guess somebody's age very well using pretty much any data type, AI can do it better using the same data type, given a lot of examples, because it never forgets. And we've published aging clocks based on microbiome, so we can use gut microflora, next generation sequencing of microbes in your gut to predict age with very reasonable accuracy, around 4 or 5 years. We can use very simple blood tests, up to 20, starting from 20 features from your regular blood test, albumin, glucose, alkaline phosphatase, urea, those kind of features, they can be used to predict your age. And my favorite data type is proteomics and transcriptomics, so gene expression and protein expression data that can be used to predict age, but also you can use this data to later identify protein targets or gene targets that you can possibly tweak with small molecules or biologics to see if those targets are causal and if they can possibly result in slowing down of some of the age-associated processes or even age reversal, and then see if any of those targets are implicated in diseases. So that's what I'm going to talk about today. And now I'm going to switch gears and talk about how can we use AI to rapidly discover and develop dual-purpose therapeutics targeting aging and disease at the same time. So in the previous part, we covered a little bit aging biomarkers, so age prediction. Now, how do we use this age prediction and many other biological approaches to identify protein targets, formulate disease hypotheses, and then very rapidly design novel chemistry and progress this chemistry into the clinic in a commercially viable way? So to answer this question, we established Insilico in 2014, originally in Baltimore, and then we scaled globally. So my second office was in Taipei in Taiwan. And then we also opened our Hong Kong office. That's where I'm based right now. We also scaled in Shanghai and also in Montreal, Canada. And our US headquarters are in New York. We also, after Russia invaded Ukraine, opened an office in the UAE. We had some R&D resources in Eastern Europe. We moved them to the UAE, closed the Russian site, and also migrated a lot of people from Eastern Europe, top AI professionals to the UAE, and we're continuing to hire in that region. But we, of course, try to avoid being in that geography right now because that's pretty terrible what's happening there. But right now, the company is over 250 scientists globally. It's a very healthy balance, approximately half-half AI and drug hunters. And we also rely on contract research organizations in Europe, US, but mostly in Asia, that allow us to perform a lot of experiments in parallel. So we basically sit on top of many contract research organizations that allow you to synthesize novel molecules and also validate them in biological experiments very rapidly. Our C-suite is in the US. And since 2014, we took a number of awards, Fierce Biotech, top 15, and several times we were in the top 100 AI companies globally by CV Insights, and top 10 in healthcare. I think that we are globally probably around top 3. In Asia, we're number 1. And the company is organized as an yin-yang of AI and drug discovery. So we have our own AI platform, and that's what I'm responsible for, and I'm the founder. So we were originally AI first company, meaning that we started as an AI company. And then once we scaled our AI, I met Dr. Ren, so Harvard PhD, 11 years at GSK. And then he helped list a company called Medicine. It's a contract research organization, took it all the way to $8 billion market cap, and joined me in 2021 in February and helped progress. Well, first helped build several teams. So we have biology team, medicinal chemistry team, preclinical team, CMC team, computational chemistry team, formulation team. And many of those team members, they actually go directly and sit in the contract research organization. So here on the bottom, you can see that we work with over 80 contract research organizations globally. And these experts, they supervise many full-time equivalents within CROs. So nowadays, the biotech industry has changed in a similar way as computers changed in early 2000s. So people adopted the Apple model, designed in California, made in China. So now what you do is usually you outsource chemical synthesis. You don't synthesize yourself using your own synthetic chemists. That is done by the various contract research organizations. And you can easily outsource that expertise, and you can also outsource biological testing. So if you are learning how to pipette in the lab, It's actually a great skill to have if in the future you're going to train robots to do that, or if you're going to supervise scientists within the CROs. But most of the time, you won't need that skill because there are trained professionals whose job is to, do that pipetting within the CROs, and also do a lot of other tasks using very expensive equipment. So you just get the data back, you send your biological material or chemistry to those CROs, and they do the rest. So it's great for AI companies because now you can basically take advantage of this hugely scalable research infrastructure without having your own staff. And you can really focus on what matters, discovery and validation. So we started as an AI company, but then became an AI-powered biotech. And this is one of my teams in Asia. This is me and my co-CEO. So we have two CEOs. As a matter of fact, I'll just go back for 1 second. You can see that we have two CEOs. One of the reasons for that is many other companies in this field, once they go from AI to drug discovery, they stop focusing on AI. Because especially once you go clinical, it's like, having kids and getting married. So no more, exploration. You start, formulating their career paths, and everything else pales in comparison. So in this case, we decided to have two CEOs. So Dr. N would focus on drug discovery, and I would focus on innovation in AI. And we work as a synergetic team. So I test the various hypotheses in AI, send him the resulting molecules and targets. His teams are testing those hypotheses, and if our AI works and is validated, for example, we validate a specific deep learning model, we implement it in software and start using it at scale and also offer it to the others. So it's extremely important to have two heads when you are an AI-powered biotech, if you really want to continue innovating in AI. Usually, again, it pales in comparison to drug discovery because most of the value comes from the molecules that you have in your preclinical and clinical pipelines. That's also where you spend a lot of money. And we raised quite a bit of money. So we raised $415 million from a group of a very diverse group of investors, healthcare specialists, financial specialists, and AI investors. Again, we're a very truly global company. And the last rounds were led by Warburg Pincus, B Capital, Prosperity 7, with participation from many other investors. Again, the drug discovery is a very capital-intensive business. Once you switch from AI to biotech, it requires capital. And here is why. So if you haven't seen the slide, take a screenshot, make sure that you watch the YouTube recording several times. This comes from this later part, target to hit all the way to submission to launch, comes from Stephen Paul's paper, How to Improve R&D Productivity in Nature Reviews Drug Discovery. Extremely important paper. If you have not read that, you shouldn't be doing biotech. And this paper shows that once the target is identified and pre-validated, and you're going from target to hit to lead, lead up, preclinical phase 1, phase 2, phase 3, it costs you approximately $1.8 billion. And that's the development cost in 2010. You know about the Moore's law in technology, in semiconductors, for example, where your processing capacity doubles every certain period. So the same is, so you've got the opposite effect in pharma. So over time, drug discovery and development becomes more expensive. So it's called the Ehrum's law. So it's the opposite of Moore's law, because all the low-hanging fruits are picked up, and you have to take more risk to get higher chance of developing blockbuster therapeutics. But on average, you can probably estimate about $2 billion per drug. There are not that many drugs approved every year. So last year, the FDA approved just over 50 therapeutics, new molecular entities, and very few of them are novel. About 35 are small molecules, and we estimate maybe 8 or 9 to be more or less novel. So novelty is pretty low, and it's very difficult. And that's after hundreds of billions of dollars spent by the entire industry. Over the past 20 years, Novartis was the most productive company in terms of internal R&D. And in the past 20 years, they managed to get 20 novel molecules approved, so novel therapeutics. So many others have approved 5 or 6. So the productivity of the pharmaceutical industry is very, very low. It's also a market of failure. So you can see that at every stage, you've got the probability of success. Discovery of a novel target usually is the prerogative of academics. So people, I hope that many of you are biologists and you are formulating hypotheses behind various diseases. Alzheimer's, for example, right now we're still struggling to even formulate the right hypothesis for that disease, not talking about how to fight it. And many hypotheses, they were erroneous to start with in Alzheimer's. After tens of billions, if not hundreds of billions of dollars spent on that dramatic disease. So disease hypothesis is usually something that you come up with in academia, and then you try to formulate this hypothesis and try to narrow it down to individual protein targets, usually protein targets. Usually those molecular switches are proteins. Of course, you can target many, many other molecular types, like, for example, RNA, or lipids, or some specific molecular structure that may be causing the disease, either upregulating or downregulating it. So that part is the most risky. So 95 to 99% failure rate at preclinical stages. And once you go clinical, you still have about 60% failure rate in phase 2. In phase 2 is when you test efficacy. You see if the drug works. So you can spend several $1 billion, well, over $1 billion by this time, and still fail in phase 2 because the target is wrong. So AI can definitely help accelerate and cost down the process. But what it can help you with in the most dramatic way is the probability of success. So we need to increase the probability of success here. Here are, by the way, the number of years, and here is the amount of money that you need to spend. So when I started in longevity biotechnology, again, over 18 years ago, more like 19 plus, I was extremely optimistic about what's going to happen in the future, that we're going to live significantly longer, that scientists are going to find many new longevity therapeutics. Around 2004, 2006, we've heard about tropomyosin being implicated in longevity. Metformin was already there, aspirin was there. People were crazy about stem cells. And guess what? Now we're in almost scratching 2023. And we still don't have a longevity drug on the market where we know that it works and people would be taking it after 19 years. And this is why. So it's very difficult to make those discovery efforts going through all those steps. And again, it's a very costly process. So most of the time, investors just won't allow you to go after, aging. They would require you to go after a disease because they would want those costs to be recovered. So, and that poses a big dilemma, and I'll explain. But today, what I'm also going to show you is that with AI, we managed since 2020 to go through all of those phases. So novel target all the way to phase 1, almost complete, fingers crossed, in under 3 years. And again, right now, we've attempted several times, and every time we succeeded to get to preclinical candidate, and now we are also in the clinical trials. Several times we've identified moderately novel or novel targets. And that shows you that AI can definitely accelerate. So when I started Insilico, what we did is we mapped out every step of drug discovery and development. So we looked at how people discover targets, how they formulate their disease hypotheses, and how they identify small molecules to be able to hit those targets. Just to be able to identify the areas where AI can perform well and possibly even outperform humans. And we also looked at some of the best practices, absolutely best timelines for AI discovered therapeutics and AI designed therapeutics. So we looked at how people discover targets. There are over 60 philosophies. I'm not even talking about individual methods, but philosophies for discovery of targets. One way is to just read scientific papers coming from top academic labs, see how the top academics formulate those hypotheses in literature, and just pick those targets from literature. Targets are usually not patentable. So unless you have novel chemical matter against a specific target, it's very difficult to patent the target and link it to a disease. Of course, you can try, but it's usually not very enforceable. So you really need to have your own molecules to be able to patent chemical matter. And that's why some companies, especially big pharmaceutical companies, they very often pick the targets from literature. They don't do target discovery themselves. They try to maintain that expertise, so they invest in internal target discovery to understand which targets are better, but they usually rely on a massive body of academic research to come up with validated targets. Very rarely they discover targets themselves. Another way to discover targets would be to use biological data, either coming from public repositories, or some pharma companies use private repositories, comparing norm to disease, for example, and looking for differential gene expression, differential protein expression at the tissue-specific level or systemic level, and formulating the disease hypothesis. Again, there are 60 philosophies. I'm going to show you some. So what we've done when we started as a target discovery company and worked with many, many pharmaceutical companies to see how they discover targets. We didn't take their data, we took their knowledge. So very often they were asking us, okay, can we use this data type to discover targets? And we didn't know. So we trained deep neural nets, provided them with different target hypotheses, and they validated. And in some cases, when it worked, we knew that that specific data type is valuable. And now we can address some of our problem with the same methodology, not the same data. And we also started looking at how to generate novel chemistry ourselves. Because if you can generate novel chemistry yourself using AI, you own your proprietary molecules instead of just screening for molecules from libraries where somebody else might own that molecule, and also it wasn't ideal for your protein target. So if you learn how to do de novo drug design, you can really accelerate drug discovery process and de-risk it. And many of you have heard about AlphaFold. Many media were constantly projecting that AlphaFold is going to change everything. We're going to basically transform drug discovery. Well, the first publication was in 2018. There are no drugs on the market. Why? Or in clinical trials, or even in late preclinical. Why? Because, again, obtaining the crystal structure on this chart, it's a one small piece of the puzzle. You need to have many, many other pieces, and you also need to be able to invest tens or even hundreds of millions of dollars to develop those molecular structures that are generated by AI into drugs. So just generating a hit, it's not even considered to be credible big step in medicinal chemistry unless you're targeting the undruggable. And what we did, we looked at all of those steps and tried to combine them in one seamless workflow where we identify targets, generate molecules, and predict the clinical trials outcomes. And in chemistry, for example, we started also using those deep fakes, same technology that I showed you that can be used to generate fake humans. we started using it to generate small molecules with the desired properties. And we came up with 3 platforms. One is called Panda Omics, which allows you to identify novel targets. I'm going to show you a quick demo on how to use it. And again, it's so simple that a super advanced high school student can use it. Chemistry 42, I haven't seen high school students using it. Even top medicinal chemists usually need training. But it allows you to use many AI models to very rapidly generate from scratch small molecules with the desired properties. So you give it a protein target, it identifies the pockets on those protein targets, and very rapidly generates small molecules with the desired properties, trying to minimize toxicity, maximize novelty, synthetic accessibility, so you can synthesize those molecules, metabolic stability, and also you want to have high diversity. And there are many other properties. Of course, you want to get a good binder. And we also developed a system called Insilico. We're going to introduce it to the market after 7 years of development because it requires a lot of validation, retrospective, quasi-prospective, and also prospective. When you make predictions, wait for those predictions to come to read out, and then you compare it with with the actuals. So that system allows you to predict phase 2 to phase 3 transitions. So probability of success on phase 2 human clinical trials using predominantly preclinical data that's publicly available. And by the way, mark your calendars. You can find, if you go to insilico.com, you can register for our launch of those 3 systems on November 14th. It's going to be epic. I'm trying to make a pretty fun show out of it. We're also going to do some really cool demos in the metaverse, again, hopefully, as they say in the Middle East, inshallah. Those systems over time acquired superpowers. So now that the entire industry started using them, we now see how other people are doing it. Humans are very creative creatures, so they always come up with new ways to use our tools. And we try to improve those tools and take them to the next level. So now let's switch to dual-purpose therapeutics. Very often, I'm being asked, and many other aging scientists, they debate whether aging is a disease. I published a number of papers on that, hypothesizing why it is important to establish this kind of paradigm to implicate aging as a disease, put it on ICD codes, so the WHO recognizes it, so we have payers who can reimburse anti-aging drugs. Well, it turns out that once you get deeper into drug discovery, it doesn't really matter, because we shouldn't even care. Even if people recognize aging as a disease, so yes, you're going to have more people focusing on the problem, because then you feel that all of you are sick, right? And you get to the peak, and afterwards it's like all the way down and decline, right? And death. So from that perspective, we all should be working on aging research. That's why I committed my life to this problem. And I'm not interested in anything else. And right now it's 2 a.m. in the morning, and I'm still preaching to you this particular concept. But regardless of whether aging is a disease, we can now develop dual-purpose therapeutics that target a specific disease, but longevity dividend, I'm sorry, it's a typo here, I'll fix it. So longevity dividend is a bonus. So you are targeting a disease with a drug, but at the same time, if you are targeting the fundamental aging pathways, the patient indirectly benefits. And you can also conduct normal clinical trials, collect the aging biomarker data using a contract research organization, for example. We again try to prefer not to touch human biological data, and see if that drug is moving the needle of your aging clock, of your aging biomarker in one way or another. So you can develop dual-purpose therapeutics. And there are examples of dual-purpose therapeutics. For example, acarbose. It's an ancient glucose modulator before metformin. Very safe. Side effect is flatulence. Yeah, you are going to maybe smell bad from time to time if you take it, but it's a really nice way to modulate glucose, and mice on acarbose do live longer. If you can combine it also with rapamycin. Metformin demonstrated some longevity extending effects in animals, and now people are trying to conduct massive clinical trials for metformin. And indirectly, meta-analysis demonstrates that diabetics on metformin may live longer than non-diabetics. Rapamycin is likely to be the most potent drug out of all three, and it extends lifespan in pretty much every model organism that it's tested in. And it's a cancer drug and also an immune modulator that is very often used for organ transplantation. However, there are very, very few people who are utilizing this drug for off-targets for aging. And of course, there are groups that are currently hypothesizing doing human clinical trials around those drugs, but there is a problem. So all of them are generic. And if you are to prove that they are implicated in aging, you would not be able to make a lot of money on it. And that doesn't justify the cost of a huge cost of a clinical trial. So who is going to pay for that? So currently, Nir Barzilai and collaborators are trying to design a study like that for metformin. It's called TAME study, targeting aging with metformin. But if you were to imagine that you are trying to identify a drug like this, a dual-purpose therapeutic, like metformin or rapamycin from scratch, so where you can actually own the IP, then you are talking about major benefit, where you can test the aging hypothesis, try to do it right away at the drug discovery stage and drug development stage, and still have the patent life on that therapeutic to go dual purpose. And there is always a balance when you're developing therapeutics between confidence and novelty. So many of the protein targets are very well known. So for example, the TOR pathway and the mTOR complex, that is the target of rapamycin, is very well known. But before you rush to develop and discover a new rapalog or a drug that works in a similar way as rapamycin and go into the clinical studies, you need to realize that it has to work better than rapamycin. And rapamycin already works reasonably well, and it's reasonably safe. So at least there is an established safety profile. If you want to go after a different protein target, so increase the level of novelty pretty dramatically, you are not going to know whether it's going to work or not. You shift a little bit to the right or to the left, and biology is so complex that the probability of failure increases. So every time you make this kind of bet, it's the trade-off between confidence and novelty. And what I'm trying to do, and that's kind of my current vision and my current passion, is to identify therapeutic targets that are implicated in aging and disease at the same time. So trying to find this intersection on the Venn diagram of targets that are implicated in both, so something like TOR, with higher confidence. And for that, we recently published a paper. So that's kind of the highlight of my today's talk. I highly encourage you to go and look at this paper, because this is the first time we used AI and openly published a method and also the targets that are implicated in both aging and disease. So we used our PandaOmics. I'm going to show you how it works, so bear with me. Where we took a lot of data sets publicly available from age-associated diseases, non-age-associated diseases, and also a lot of data sets from healthy patients. Usually it's an adjacent tissue from a person with a disease, or it's a tissue sample from a healthy volunteer that was used in a big study, and then the data was published. So we utilize omics data, so different gene expression, methylation, and protein expression profiles. We also combine it with text, so we integrate a very large number of publications and abstracts of publications, also figures of publications, and we also monitor biomedical grants. grants are published before publications and very often do not result in publications at all if they fail. We also monitor key opinion leaders, so KOLs. We try to establish how trustworthy they are, because very often scientists make mistakes. And we also monitor financial information coming from biotechnology companies, publishing press releases and other kind of reports. And then we process this data all together, try to establish target lists like this one. I'm going to show you a demo again. And we try to establish druggability, development level, tissue specificity, safety, and many other properties of a target. And we originally usually start with low novelty targets. So here you can see novelty column. So most of the pharma people will be familiar with those targets. And then you can gradually increase the novelty and still retain confidence. So, and then we identified a bunch of targets that are common in multiple age-associated diseases and healthy aging, and mapped it on hallmarks of aging. So we came up with this target wheel. I think that's one of the most important publications in my life, probably, because here we used AI to map the different targets. Many of them are very well known, and structured them by their implication, how implicated they are in specific hallmarks of aging: cellular senescence, extracellular matrix, genomic instability, et cetera, et cetera. There is a very famous paper, which is called Hallmarks of Aging, which describes the many areas that are driving the aging process. And we also structured those targets by confidence and druggability. So if you are a venture fund, if you want to go for something novel and you have a lot of capital and you can risk, you can go after highly novel, medium druggability, or high or low druggability even targets. Usually you won't do that. So usually you want to get a healthy mix of something novel, something medium novelty, medium druggability, and medium novelty, high druggability. If you are starting from preclinical level. And then some of those programs will definitely fail, but some will work out and pay for everything else. So that's kind of the mantra in drug discovery. And right now, you can imagine that I am pretty connected in the industry. I speak at 80 to 150 events a year. And I'm connected with many venture capitalists. Very often they use me to get advice. And right now, if you are getting into this industry, this is a very good mantra to build your own pipeline of therapeutic programs. Either pick a few targets and start developing your own drugs, or in licensing the small molecules from big pharma to repurpose them towards a specific age-associated disease indication, and also go after aging. A company called BioAge, led by the wonderful female entrepreneur, also Canadian, who started a company called BioAge, and they are repurposing, and currently they are running 3 phase 2 clinical trials. Amazing company. We decided to go the discovery way. And we now want to democratize and demonetize drug discovery. So we introduced a lot of very cheap tools. So one of them is Pandaomics. It allows you to identify protein targets. I'm going to show you just one demo on colorectal cancer because it's just easier to comprehend than going after chronic disease. So see, we start searching for colorectal cancer. It shows you 811 data sets that we have from public repositories. Those are already processed. We had a lot of humans see which one of them are clean, which one of them are not. So you can see that we monitor here about $6 billion worth of grants, 16,000 grants, 5,000 clinical trials, just over 100,000 publications. For a human, it wouldn't be possible to go through this information quickly. For AI, it is possible, and you can see that related data sets are structured by data set name from a public repository. So most of the time, it's National Library of Medicine, it's GEO, Gene Expression Omnibus, something that you work with all the time. Here are the patient samples. So number of samples, patient samples, controls, usually healthy controls, number of attributes associated with the data set. Usually it's age, HPV status, et cetera, gender, sex, gender is optional. There is a platform and when it was published. And also it gives you some basic competitive information. So how many unique drugs are there, how many small molecules, biologics, which stage are they in. Then you can select from this large list of data sets, the data sets that we kind of consider more or less high confidence with a large number of samples. And you can do basic quality control. So you select the cases in this data set and disease samples, so controls and disease samples. Do the very basic primitive bioinformatics, so do differential expression of genes, right, to see if this data set kind of fits your hypothesis and is more or less clean. Then we group genes into networks, into biological pathways using our IPANDA algorithm. So IPANDA is Insilico pathway activation, network decomposition algorithm that we published on Nature Communications in 2016, but we came up with this algorithm a while ago. Allows you to do dimensionality reduction and look at differential state change in signaling pathways, and also identify which nodes contribute to either activation or downregulation of a specific pathway. So it allows you to make some of your AI work that I'm going to show later more interpretable. And then you create a data set of, let's say, 3 data sets like this. So you combine them and you get to this interface. And then you are going to continue working with this interface. I mean, on the new version that we are releasing on November 14th is going to be pretty dramatic changes. But here you have a number of genes that encode proteins. They are ranked by omics scores. So there are several AI engines using different philosophies that rank those genes using gene expression, protein expression data, network topology, and many other approaches. Then we also look at how much evidence do we get from text, because here those are low novelty targets. So novelty is red. So we use a traffic light system here. And you can see that many of them are not accessible by small molecules or biologics, and some of them are not exactly super safe. And so basically it gives you huge executive summary on target space. And usually the pharmaceutical industry executives, heads of the therapeutic areas, they're like, oh, okay, I know this one. And that's actually why we do this exercise first, showing low novelty targets, because it gives more confidence in the system to the pharmaceutical industry executives, and also kind of gives you more confidence that you are doing the right thing. So now we're going to prioritize the targets that are more accessible by small molecules. So we want to make this column green, again, to impress the big pharmaceutical companies head of the therapeutic area. So they believe in the system a little bit more. And also some companies that do not really need a lot of novelty, but want to have some preclinical pipeline assets that target a specific disease. They can pick some of those lower novelty, higher drugability targets. But now, if we want to increase the level of novelty, we will reduce the reliance on text data, on prior knowledge. So we will try to deprioritize text, and we're left with this interface, which provides you with higher novelty targets. So now the novelty index is green or yellow. They're accessible, those targets are accessible by small molecules. There is very little evidence from text and financial reporting that anybody is working on these targets in the context of colorectal cancer, and you can just start testing. So that's how we do target discovery, just for cancer, for example. For aging, it's much more difficult, and there is an additional step that we perform. By the way, if you want to see how to do target discovery at high school level, I encourage you to take a screenshot here, go to Inspire Longevity breakout launch and find it on YouTube, and see how 2 high school students, amazing guys, Andrea Olson, 17, and Zachary Hapa, 16, how they used Panda Omics for a year, came up with disease hypotheses and also target lists for glioblastoma, and also some of the age-associated targets that are implicated in glioblastoma and aging at the same time. And when they were presenting at ARDD in Copenhagen, they actually got a pretty substantial audience. So most of the people went outside for lunch, and they were running back because this guy started the show, and you can see those wonderful diagrams which show those targets with low and high novelty. So please do watch how high school students do this, and basically it shows you that you can do that yourself as well. If you have a good principal investigator that is willing to work with you, a KOL in a disease, in a specific disease, you can utilize this tool to come up with really interesting target lists and hopefully validate, publish, and develop a drug. So, and watch those guys. If you are a PI by any chance watching this, you should consider getting these guys for an internship and then getting them into your lab. So we really hope that the next generation of scientists who grow up from those high school kids will be able to go after more ambitious projects in aging. So now I'm over time, so I'm gonna skip a few slides. I hope I'm not gonna make you slide sick. I'm just gonna show you one fun study that we recently performed in ALS, amyotrophic lateral sclerosis. A lot of you know it through Ice Bucket Challenge. So thank you, Mark Zuckerberg, for popularizing. A lot of AI companies decided to prioritize ALS for research because it's maybe a little bit faster to conduct clinical trials, and also there is a lot of attention to this disease. It's age associated, so average age of onset is 55 to 65. It seems rare, but there is a lot of people who even I know personally who got ALS. And we used our Panda-omics in a similar way that I just demoed to identify promising targets using multiple public data sets. So first, we went through public data sets that I've just showed, integrated from Geo, AreaXpress, and several other repositories from ALS patient brain sample. Then we also worked with the Answer ALS consortium. They also have a public data set of really high quality data, but they use iPSCs from patients. So they take a tissue sample, reprogram the cells back into the baby state, and use that as an ALS model. So again, what is better, brain samples from dead people that have degraded quite a bit, or reprogrammed iPSC cells, we don't know. But here we try to combine them, used Panda-omics to identify novel targets, and then we also looked for drugs that you can repurpose. And then we used a model from Ke Zhang, at that time at Mayo Clinic. He developed a fly model of ALS. And by the way, right now, there is no good model of ALS. So you cannot really experiment on people right now. I mean, you probably could, but they are so diverse, and it's better to establish the target hypothesis first. So there are some mouse models. Most people use organoids. Some of our competitors use organoids that they develop into kind of quasi-brain tissue and then test the drugs on them. But here you've got a live animal, and you can also test some of the hypotheses in their neurons connected to the sensory system. We also collaborated here with the Answer ALS founders, so Mary Chitkovitz, the head of neurology at Harvard, Jeffrey Rothstein, head of Answer ALS, head of neurology at Hopkins, also Bailu, a very famous professor from Tsinghua, and Ke Zhang from Mayo Clinic. We published those targets, and we validated some of them in flies. So the targets that we predicted using AI were validated in flies. And you can see that some of those RNAi experiments, when you're doing target validation, had strong rescue effects on those G4C2 upregulated population model. And we've prioritized several targets that were more or less validated using this model. Again, we did a quick and dirty study and published and wanted the others to validate. And of course, we are also looking at some promising targets and looking for additional, possibly charitable funding to prosecute some of those targets. But I have a colleague. She's a wonderful computational chemist. She very rapidly progressed from leading our Chemistry 42 to the head of a platform. She also runs our Montreal site. Her name is Katrina Kamia. She told me, like, look, Alex, we are presenting at a conference that was last week. How about we pick one target out of the ALS study, challenge our chemistry team, give them this target for a weekend so they can play with it. So at the medicinal chemistry conference, we can, show some results. And I said, oh yeah, so go motivate those guys, right? Because they're already overloaded. And she decided to give them this task. So a couple team members were up for this task. So they started looking at which targets are more promising in this list that we've published. And we, for the first time, utilized Insilico system, which also prioritized 2 targets for prosecution that have more or less, again, those are not ideal scores for success or failure, predicted success or failure in clinical trials using our Insilico platform. But we prioritized NRC1 and ADRI2B, adrenergic receptor, for prosecution, and decided to do this experiment first using our Chemistry 42 that allows you to generate novel molecules with the desired properties using generative algorithms. Usually, you would go through a very extensive process, multiple cycles of hit identification. You identify the hit, give it back to Chemistry 42, identify, improve different properties, and then you confirm the many properties in animal models, also efficacy, and nominate preclinical candidate. So here we decided to do a quick and dirty study. We decided to look at already existing molecules for NR3C1 and give it to Chemistry 42, both the target structure and the resulting molecules. And the Chemistry 42 very rapidly redesigned those molecules so that medicinal chemists, when they look at those molecules, they like them. And the beauty of this this exercise was that, and that's when you show those molecules that are generated and you show them to medicinal chemists at the conference, they're like, yeah, I would probably have done these things differently, but this looks promising. So for them, I'm not a medicinal chemist. I think that medicinal chemists are probably the smartest people on the planet because they are like the MMA fighter of everything. They can do biology, chemistry, drug discovery. They usually are super expert. And they also understand the language of chemistry. When they see something like this, they are like, "Oh, okay, this is good," or "This is toxic." And that's one of the ways to impress chemists at a conference. And that's also the output of our Chemistry 42. We can now synthesize and test. That's the next step. But it doesn't. So people will not believe you if you haven't validated experimentally. So only phase 2 complete is when you can tell yourself that the drug is more or less a drug. Of course, phase 3 is the ultimate test. But here is a question. So what did I do to date in experimental validation to show that dual purpose therapeutics have a future? So in 2020, we decided that we are now so well positioned in AI that we can go after our own drug discovery program. Novel target, novel molecule, broad disease, no cure, associated with aging. So think about this ALS case study that I showed you, but a real program. We decided to prioritize fibrosis and aging at the same time, and also idiopathic pulmonary fibrosis. Again, age of onset is mid 50s, 60s, so age associated. And it kills very quickly. So patients usually have 2-3 years, terrible death. To help them, we decided to look at, we developed a deep learning engine, which predicts age and fibrosis status at the same time, looked at common targets, fed this output to PandaOmics, prioritized 20 targets. We could test only 5 because for the rest, the protein was not available. So we tested all 5 worked. And one we prioritized for multiple indications. So we nominated the preclinical candidate. Usually, again, to nominate the preclinical candidate, you need to perform a lot of experimental studies to demonstrate that the target is efficacious. And we've demonstrated that in lung fibrosis, kidney fibrosis, almost done with skin fibrosis. This is for the data package that you need to nominate the preclinical candidate for just IPF. So we even did lung function assays. So put mice in some containers where their lung capacity is, lung function is measured, and you induce fibrosis, try to rescue this fibrosis with a drug. Also, we used donor cells and many other assays. So nominated the preclinical candidate, and we did it under 18 months. So that's a pretty fast. So to my knowledge, that's the first target discovery exercise with chemistry that went to preclinical candidates so quickly and on such a small budget. Then we performed many additional studies called IND enabling studies that allow you to go into humans. So 28 dog and mouse studies, so no major side effects observed. And we started the phase 0 study because the target is novel. I was really worried about starting a large-scale phase 1 study. And last November, I decided to do a small 8-patient study in healthy volunteers in Australia. So a contract research organization performed it. We didn't see anything undesirable and decided to start the phase 1. So to my knowledge, that's the first time where AI was used for target discovery, small molecule chemistry, route planning, and we went all the way into phase 1, now waiting for this phase 1 to be complete. And again, I think that this is a dual purpose therapeutic because we identified it using aging research. So hopefully we will be able to, after we demonstrate efficacy, again, fingers crossed, in fibrosis, we will be able to repurpose it for aging using the standard data that is used, that is generated using a clinical study. So we have not disclosed this target, but as I've mentioned, we have 31 programs, and some of the programs I can legally disclose. So some of those programs, like for example, PHD2, it's HIF1 alpha. It's a target that is also implicated in modulation of erythropoietin, EPO, and BioAge, the company that I've mentioned before that is using other data types to identify targets and in-license molecules, they also found with a completely different approach, HIF1 alpha PHD2 as a promising candidate target. So we prioritized it for anemia and also IBD, so inflammatory bowel disease, and there it's a gut-restricted molecule. It doesn't get into the bloodstream, so very specific design. And are also planning to go clinical. This is a little bit older target. We're trying to make just a better molecule than what's available right now and completely novel. And also another really fun target that we've discovered and implicated in immune oncology, but we also believe that it has a role in aging, is QPCTL. And we signed an agreement to co-develop it with Fosun Pharma. It's a pretty substantial innovative pharmaceutical company that also gave us 50% rights in our own target that we've discovered, gave us some really nice upfront, and we've already nominated the preclinical candidate and are preparing to go clinical. So, and many others are for sale, right? So you want to make great business and return nice returns to the shareholders. So that's why we have to go after hot areas like synthetic lethality with targets like USP1. That target is actually, major contribution was done by David Sabatini, previously at MIT. So there is a company called TSQ Therapeutics that is leading in this area by probably 6 to 9 months. And we are the second, so fast follower. We really hope to make best-in-class molecule. And some molecules like MAT2A, it's the modulator of regulator of SAMe, so adenosine methionine. So it's a protein that kind of gives you methyl groups that is implicated in methylation. So if you inhibit SAMe production, you don't have remethylation occurring very continuously, right? And that's a way to study methylation and methylation aging clocks. Many of you have heard about methylation aging clocks. And by the way, it's a promising cancer drug as well. Several pharma companies are pursuing this as well. But again, for us, we try to go as dual purpose as possible, but also maintaining commercial tractability. And since 2021, we nominated 8 preclinical candidates. As I mentioned, many of them are dual purpose. And now I'll tell you a very fun story in like 2 minutes or less. Hopefully, I know that I'm over time and we want to leave some room for questions. But now, since we have already exhausted those massive publicly available data sets and our AI is already trained, we could, in theory, benefit from additional incremental data that is proprietary. And we are originally planning to use animal data because that's what's lacking. And I'm launching my fully robotic AI-powered lab where no humans are allowed. I hope to do that in December. So I'm going to go to our lab end of November. This is kind of the sketches with some field application tests of individual stations. So it's a lab which relies very heavily on those small robots called AGVs, autonomous guided vehicles, to transport stuff. For example, microplates with compounds or the cells or organoids from incubator to the high throughput screening station. So humans do not go into those rooms. Every island here is automated. And in between the islands, we have those single arm and dual arm AGVs. We had to design our own and also work with a company called X Imaging that is specializing in those robotic solutions. So in that lab, we're going to have an island for compound management, cell culture, and NGS, high throughput screening, and cell imaging and analysis. So we are trying to focus on deep phenotyping, so high resolution imaging of cells and tissues, transcriptomic data, proteomic data, and methylation. So those are the kind of 4 data types that we want to generate and combine them. So all of those 4 data types come from each individual sample. And then we expose those samples to a large number of predicted molecules predicted by our AI. And then we measure response on all of those 4 data types at the same time. And again, it's done in a completely automated way. I'm going to skip a little bit, right? So because those are like typical robotics that you see in the lab. So it's Hamilton liquid handler. We also have standard echo screening equipment, which allows you to perform many experiments at the same time and run a number of reactions. We use a standard Illumina NGS sequencing machine, but in order to process the samples, we use those dual arm robots at the station that allow you to do high throughput tasks, but also do precision pipetting, for example, and perform multiple experiments right away from niche methods. And you can do this in a very consistent, standardized manner, and not being worried that a person forgot to do RNA extraction mix properly or prepare the gels properly. So it's all automated and consistent. Again, originally we were going to start with animals because I actually feel that there is a lot of animal data sets lacking. A lot of people are focusing on humans, and that does not allow us to have good translation. So for the first half year, until we get any specific permits, we are not going to use human data. So here is my contacts. Very happy to answer any questions. And again, we're truly global, and we are focused on one goal, delivering best therapeutics to patients in need, preferably with dual purpose. Happy to answer questions.
Thank you so much for giving that amazing talk. Lots of food for thought there. I've got a question, and if anyone else has any questions, feel free to pop them in the chat because this is live streaming on YouTube, and if you don't want to broadcast your voice or your face, then I can just answer your question. I can say your question. So I'd like to kick things off with my question all the way from the beginning of your presentation. So for the slide on the aging clocks, right? So looking at factors like the deep micro photo aging, how do you weight the different factors, and do you combine all of those factors into one like generalized aging factor? Maybe like have you gone like a step further into that?
So it's a very good question. So to my knowledge, the Deep Longevity Group published the largest number of clocks to date, right, on many, many different data types, ranging from, pictures of the face, publicly available, the data available from wearables, and also gene expression data from public repositories, blood tests, etc. So every one of those aging clocks tells you a different story, because your body is composed of many cells, many tissues, many organs, and each one of those levels of our biological organization has a clock. So you can measure, racemization of amino acids in proteins. That's what I did for my PhD. And that's actually an aging clock by itself, so very often used in forensics. So if you find a body somewhere in the wood, preferably animal, if you want to evaluate how old it is, you can take alpha crystalline from the eye, or osteocalcin, or some other substance that contains a lot of long-lived proteins, and measure the ratio between left and right amino acids. For example, aspartate or asparagine, they racemize a little faster. And using this ratio, you can guess the age of that animal. Is racemization contributing to some diseases? That has not been established. So there are some hypotheses that when it happens in the eye, you've got, that's one of the reasons why you don't see very well. And maybe that's one of the reasons why those ultra long-lived organisms like bullhead whale, lose their sight over time, even though they don't age as fast as us. Some clocks, for example, methylation aging clocks, they can very accurately predict your age, and they are associated with diseases and general fitness and frailty. Many studies demonstrated that, and they might be affected by smoking and other epigenetic factors. But those methylation sites that are most predictive of age, they don't sit in promoter regions of any of the known protein targets. So they're not exactly great for target discovery, but they tell you a story. And in my opinion, those clocks actually measure differentiation more than aging. So if you take some kind of less mature cells and extract them from your bloodstream, they are likely to be much younger. So when you're doing bulk, then you get very accurate readout. My favorite data type is transcriptomic data, so gene expression or protein expression. Gene expression because there is a lot of gene expression data available in the public domain, so you can train on this data. And you can also reasonably cheaply measure it if you are to validate. And you can use it for target discovery. So if you see that some genes are much more important than the others for the accuracy of the prediction, so imagine you train the deep neural network with many, many input neurons, each one corresponding to an individual gene, and just one neuron on the output. You can try to see if you can randomly shuffle each one of the genes going in sequentially, and create a histogram of the importance of this gene for the accuracy of the entire predictor. And that would be one of the primitive technologies to kind of make those deep neural networks interpretable. Find some of the most promising genes and see if they have any biological relevance, and they could be possibly related or implicated as a target. So that's my favorite data type. Very often those clocks are not ticking in the same direction. So for example, if you are evaluating the skin aging clock, right? So by the way, you can do it using many technologies. One way is to use photographs, high resolution imaging, low resolution imaging. You can use gene expression from skin, and many other clocks. But imagine that the patient was sunbathing or they're a truck driver who likes to have the window open when they drive. And on one side of the face, you are going to have accelerated aging due to constant UV exposure. Is this and the aging clock will show that subject as an older individual from one side of the face. Is this biologically relevant in the context of longevity? Are they going to live longer? Are they going to be more susceptible to skin disease? Well, maybe not. So again, every clock tells you a different story, and usually you want to design a clock for a specific purpose. We try to combine many of those clocks together, and usually then you also are doing it for a specific purpose. Usually you're trying to see if the combination is more predictive of specific of mortality, of overall mortality, or specific cause mortality, or the onset of a specific disease. So we see that some combos are more predictive. And very recently we did a fun study. So we, using Charles data, it's the China Health and Retirement Longitudinal Study, open data set that is openly accessible. There are many surveys that you can use to predict the person's psychological age. So basically, you can ask a bunch of questions to a person and train the neural network on many of those answers to predict the person's age in a healthy state. So we published on that several times, and then some of those data sets are also connected to blood tests that are openly accessible, and you can train the deep neural networks on that blood test data to come up with the blood age. And what we've seen with this Charles data set is that some of the psychological features, like for example, loneliness or optimism, affect your biological age in a very dramatic way. So that study was published 3 weeks ago and it went viral. So, Fox News, Fortune, everybody covered it. Most of them did not really cite the paper, unfortunately. So, but it's still, it has very high altmetric, so the measure of public attention. And the title of most of those stories is that being lonely accelerates aging faster than smoking. So again, some of those clocks, like psychological aging clocks, are very useful to just establish the various methodologies for testing those clocks, because in psychology, you can actually manipulate those clocks and try to reverse them. For example, if the person believes that they're going to live for 150 years, well, no, they're going to live longer than before, than they were planning to live before, they start behaving as younger individuals, and they also answer questions, follow-up survey questions differently. So psychological aging is plastic, as shown and demonstrated by Laura Carstensen out of Stanford. So she is the world authority and the founder of social emotional selectivity theory, which kind of postulates that the shorter you expect to live, the older you're going to behave. So even some of those psychological aging clocks are great models for studying aging clocks in general, because some of the results are easily interpretable by human intelligence. So you can look at some of those survey questions that are most important, that are driving your behavioral pattern, and say, oh, okay, well, I know why, right? So yes, we've seen that before. So if a person is lonely, yeah, they're aging faster, and most likely they're gonna die sooner. So they're also very correlated with mortality. And one way to address that is, okay, be a more social creature. Find a partner, find a friend, or convince yourself that you are going to live longer, right? Because longevity optimism is another very important feature. And once you validate some of those feature selection hypotheses with human intelligence, you can go to biological aging clocks, and they become much more interpretable because you've already validated some methodology on something that is much more interpretable. You can also do that with imaging data, by the way. You can make pictures of the skin and identify specific features that you can interpret as a human, like wrinkles, for example, or dryness, or sebum, or translucency. There are many, many other features. And once you establish the algorithm for feature selection, you can go to more complex data types like transcriptomics and see if some of the feature selection methods that you've validated on other data types may be working on transcriptomics. I hope I answered your question.
Yes.
The easy way to answer would be, yeah, we did that.
Yeah.
And of course, that's the holy grail of aging research. Yeah.
Yeah, thank you. You gave much more information than I expected, but I feel like I got a very deep insight into aging clocks from your answer. So thank you very much. Any more questions? I think I'll just take one more question if there is one. If not, I think in the interest of time, we should finish the call. Any more questions? No.
I see that there are many questions in the chat.
In the chat. Okay. Yeah. Sorry, I was not looking at the chat. Okay. Should we? Ooh, okay. Oh God, that's quite a lot of questions. Okay, could you answer them? Yeah. Like briefly.
Sure. So can we regard to multi-specific drug technology while mentioning dual purpose therapeutic? Great question. So currently, of course, I do not subscribe to the philosophy that, you know, there is only one target, one disease. Usually there are multiple targets driving many more processes driving the disease and aging as well. So yeah, you cannot just go one target, one disease, but if you want to have a clean kind of FDA approvable study, something that you want to pitch to the regulators and you know, demonstrate that technology works, I think that you need to start with one target, one disease, and demonstrate that your algorithm can pick targets for that specific purpose, and also narrow down the population where this target is extremely important, or more important than other people. And then, of course, we need to go multi-target.
I have one question that was sent to me directly. What do you think of autologous versus allogenic therapies? Which categories do you think will be the winner in the long term?
Well, I've spent a couple years in a pediatric oncohematology center where we've studied bone marrow transplants and other cell therapies. I think that both technologies have the right to life. And in some cases, you would really need to go allogenic because, you know, it's just speed, or if you're looking at, you know, skin grafts or something where graft versus host is not going to be a huge problem. And also you can clean up some of those cells from anything that is immunogenic. I think that both technologies are promising, so I would not prioritize one over the other. I would just look at application-specific tasks. As a matter of fact, you know, one crazy idea, completely crazy, if you are to go after something that is not only moonshot, but probably sunshot, because you're going to get burned in the process, would be therapeutic cloning. So think about cloning a body with kind of a clone that doesn't have higher high neural activity, right? So they maybe pretty much think of an animal with your body parts. And I think that's the way to dramatically extend lifespan, right? If you had a source of young, healthy, completely autologous organs, that is the true holy grail, right? So then you're just at the mercy of your brain aging, and that's going to happen slower if the rest of your body is younger. So yeah, I think that would be the most promising technology, but of course the ethical aspect of that and also the technological threshold there is pretty high.
Another question, what do you think is the role of small biomedical university labs in the face of AI drug discovery / fully automated labs that can often produce much better hypotheses than the standard approach?
I think, again, both have the right to life. So those small biotechnology labs, if they are managed by superhuman and also using AI, they can be extremely effective. And many of the experiments you just cannot automate. You really need to develop the assays in a very specific controlled laboratory conditions, and you don't need to scale. And many target hypothesis generation methodologies require just a few simple experiments. So you don't need to have this scale. This scale is necessary when your AI gets much more data hungry and when you want to produce massive data sets for machine learning and also combine those data types, for example. That's where you really want consistency. In some cases, you can discover a target by using just one patient.
Cool. Another question. How could a mathematician who works in the theoretical probability theory and analysis, but has no knowledge of biology / chemistry and only basic programming skills, get into this field? And what would be the best first steps in this case?
Great question. So if a high school student can do it, a high-level mathematician can do it probably better. So I would start by learning biology first. And there are plenty of Coursera courses that allow you to get this knowledge in a minimally invasive manner. So I would start by learning systems biology first, because that's kind of as close to math as it gets, because many of the processes are pretty algorithmic, and it's memorization and just basic understanding. So I would pick up Lodish's Advanced Cell Biology and start reading, in addition to a few Coursera courses, so Introduction to Cell Biology. I would also take a few of those courses at longevity.degree. There it's super primitive, right? So we try to go to the level of the most kind of simple, basic medical training, right? So many medical professionals outside the US and the UK do not get a lot of electives in biology. So you need to make it kind of common denominator. And there we just explain in a very similar way as I presented today, the basic biology of aging. So you learn. Then I recommend attending a bunch of conferences. So like ARDD, for example, in Copenhagen, so this conference. And there are lectures that will be accessible to pretty much anybody. You will also meet some people who are not exactly hardcore biologists, and you can work on a team on a group project as a volunteer, and then maybe start doing that as a career choice.
Oh, thank you very much. And one final question. So from Shimon, his question follows, how do you maintain mental clarity at 3 a.m.? Like, can you tell us a bit more about your sleep routine, and especially when you travel frequently between multiple time zones?
It's motivation. So I just think that this is the most important thing to do. And, you know, if one of you guys starts performing well in aging research or just prioritizes that and achieves, you know, really nice results, you know, life is well lived. So you never know. Like, for example, those high school students that we've met, you know, for the first time, I actually met them at this conference. And I already before for 2 years, these guys were communicating with my team remotely. And I actually think that the way these guys are formulating their career is pretty dramatic. So it really opens a lot of opportunities for like the internet allows people to learn in completely new ways. I hope that metaverse will allow us to go even further. And I just think that aging research is the most important thing that anybody can pursue, because a good physician or a good surgeon generates, in my estimates, 24 to 30,000 quality adjusted life years during their career by quote unquote saving lives, right? So adding a few quality adjusted life years by fixing problems. If you discover a drug that adds 1 year of life, one quality adjusted life year to everybody's life, you generated 8 billion life years. So, and that's not such a challenging task when you think about just 1 year, but for everybody. And if anybody up there is counting, that's the thing to do, right?
I'm so sorry. One last question. Okay, this is definitely going to be the last one. So where do you think the big breakthrough will come from in aging? Like, do you think, I don't know, I find that like a very wide and vast question to answer in like a minute or two, but
well, I can tell you where it's not going to come from. So what we do with AI-powered drug discovery, I think that, there we can probably add, as they say in the Middle East, inshallah, we can probably go 10, 15 years, right, with pharmaceutical means, maybe slightly more. But I think to get to the next level, you really need to go regenerative medicine. And I think that the big breakthroughs will come from tissue organ transplantation.
And I think definitely the new research on organoids and definitely like trying out those drugs on there is going to be a big sort of leap forward. So yeah, so thank you so much.
But drugs, pharmaceuticals, guys, once you get deeper into the field, you realize that first of all, the process is very long. We're talking about many years or decades, huge probability of failure. Very few of them get approved, and very few of them you will see discovered and developed in your career. And the probability that it's going to also extend life is pretty low. So once you get into the field, those rosy glasses are very quickly removed. You lose them, right? So it doesn't mean that I'm not optimistic. It's just there need to be other technologies that will dramatically extend our life. And most likely it's not going to be pharma. It's extremely important to start in pharma because if you have a few success stories, you actually recover all of your investments and you make a lot of money that allow you to do other research. And of course, you develop a lot of enabling technologies, but I think that regenerative medicine is the next big thing.
Okay, sure. I think that will be the end of the Q&A session for this talk. And again, I just want to thank you so much for coming to talk at our society. I'm sure everyone in this call was fascinated by everything that you had to talk about today. Personally, I really enjoyed it, and it feels like science fiction brought to life. So thank you so much. And
great pleasure, guys. And my recommendation, get Christian Fortney from BioAge to present to you. That's a really, really cool speaker.
What's his name? Christian
Fortney. It's her. So she is a wonderful scientist out of the University of Toronto and then at Stanford, postdoc there, and started a company called BioAge. So they are technically our competitor, but it's an amazing company.
Okay, I'll definitely connect with her on LinkedIn and try to get her for the next thing. So yeah, thank you so much. And I don't want to keep you from your sleep any longer. So thank you so much.
No problem, guys. Yeah, thanks. Bye-bye.
Okay, thank you. Bye.